Hello. It's only been 10 minutes. That doesn't make any sense. How long to overlap? Hello. Shaky, how's it going? Can you hear me?
Hey, it's going really good. How are you?
Good. Okay, so there's no pressure. I'm glad you're here early. Did you do some algal mining by any chance? Hello?
I can't hear you.
Can't hear me?
Did I?
Mm,
Hold on a minute.
I don't know if
Okay,
it's
I
my
can
mic.
see you.
Sometimes audio. Hello? Can you hear me?
I'm here.
You
Yes.
can hear me, can you? Can you hear me?
Yes. Can you hear
Oh,
me?
yeah, I can hear you. Yeah, shoot. Did you have a chance? I guess you did look at some algal stuff, yeah?
I did. I I didn't find anything. I didn't find any other space studies. I only looked in the NASA database and I
Perfect.
found only one space study on algae, and that was Dr. Settles.
Perfect. Okay, great. So no pressure, but in theory, Dr. Settles is going to be turning up in a bit today. Might be a bit late because of he's driving through Silicon Valley, whatever. In addition, Katie, his like postdoc who worked on the space algal thing, will also be turning up. Yay! And there's probably more. How to put it? They have probably done more than is currently available through the OSDR. So the fun thing is for you to just kind of present what you found through the Open Science Archive, because everyone wants to know what access that kind of provides. And and then we'll have some really nice subject matter experts that can answer questions. And the key to it is just have a fun, lighthearted conversation. And this is part of the networking game. I hope you like it. Hopefully they should turn up. I saw him last week at
Okay.
GSR, the American Society for Gravitational Space Research. So I was able to chat with him in person. And coincidentally, Rachel Rivero, she comes to this club. She had been working with Katie, his postdoc. And they're both really lovely, friendly people. So don't worry. Just be yourself, have fun. But I kind of wanted to learn a bit more about algae, so I got the NASA experts on board to join the conversation. Hopefully that sounds exciting to you. Right.
Hello, Richard. How's it going?
Oh, hey, how's it going? Yeah, we're a bit early today, but that's all good.
That's
I
right.
was just getting Shankia telling her dreams have come true, and she's going to meet NASA's algal researchers. But
That's good. I brought the soil sensor today for a demo, so
good. We'll hopefully have some people from the regolith focus group as well. I mean, even yesterday, Ellison Blancafor from he's one of the NASA chief science officers in like the Playa. He turned up for the regolith meeting, so hopefully he'll turn up today. But it's always really, really informal. So we should probably get the link document out so we can write some notes in there. Because basically we have like an agenda which is kind of made ad hoc as we go, and then kind of helps us roll people's questions and thoughts into the ad hoc discussion. Some people like to have these things being really official and structured, and I'm a bit more free form. Okay, cool. So what I was looking for is the OSDR. M is coming too, great. The OSDR gender OSDR on AWG. Hello, Emma. How are you today?
Doing well. How are you, Richard?
Surviving somehow. They would have put one foot in the other. You know my melodramatic style. See, I'm consistent, if nothing else, right? I always use that same line. Come rain or shine. Okay, where's the chat? Trying to get organized. So you made it back in one piece from the conference, eh, Emma?
Made it back. Trying to get stuff done. Suddenly realize how little time there is before I leave OU and how many things I need to get to people before I disappear.
time flies, especially when you're having fun. So you're clearly enjoying your time there. I guess that's the best way to look at it.
Yeah, something like that.
Hello, Rachel. Good to see you. It was nice to see you in 3 dimensions at the.
I know. Yeah, it was so much fun. I had a very, very great first ASGSR.
That was your first one, was it?
First
It
of many,
was,
right?
Yeah, getting to follow Joya is I always say personally, I always say an honor, but it could be a bit nerve-wracking.
Yeah, I wasn't looking forward to that part. Because it
and
was
also-
originally supposed to be someone else presenting, and then I guess she took over, so I saw the schedule change to her, and I was like, oh no.
I switched sessions right before your talk, so in my
Oh,
mind, you were the
wow.
very first talk in the session, and it was great. No comparisons.
The other thing is it's not just like the presenter, but it's also the subject matter that you were following, which is strawberry, right? It's such a charismatic piece of food, right? So so think about like in entomology terms, let's say you're like the insect category, right? And there's you're giving a talk, and the talk before yours is on bees. No matter what species you're working on, no matter how good the science, everybody loves bees, and so it's always going to be this weird emotional thing that's already won the previous talk. In a similar way that the strawberries can do with plants, because people love strawberries. Like
That
kind
is
of
very true.
tonic.
Sorry, I'm picking off the note takers.
You kick them out. Oh, brilliant! Thanks a lot.
Yeah.
Appreciate it. Oh, hello, XYZ. I do have to ask who you are. Hello. Not that I blame you. Oh, geez, that's
XYZ.
XYZ. Yeah. Who are these? That's okay. You can also remain, I guess, anonymous. I guess I shouldn't worry about it too much. But I would always go for the quickest options too, often when logging in. In S, I'm guessing that's Ryan Scott.
Yeah, that's what I assume.
Is he on the download? Is he sneaking in? Because wow, like so. It is a new brave new world, isn't it? Like so I know that you're kicking these two, but we're having more AI bots logging in almost than humans. Right? It's close, maybe not 1-to-1, but
I'm going to quickly check the link on the OSDR calendar because every single meeting, it doesn't work and then it's fixed. So I might be joining twice. Let's see.
Oh, okay. I appreciate you checking that. I see Ryan's coming in for his phone. That's why it's not showing his normal self. Okay, Justin's here. Okay, cool. And Mark will probably be in that same traffic jam that Ryan's in on his way to work too.
I see.
He said he would like. I did get him to say.
Starting at 2.
Lindsay's here. It's good. Wonder if before we get started, Lindsay's different. Hey, Lindsay, like at the ASGSR conference, one of the students had taken those spectrum stickers and they'd like scanned the variants and such in them and compared it to like industry standards. These Macbeth charts that people are using, and the Macbeth charts were more like variable and faded quicker, and were like $90 a piece. So
Wow.
it was really interesting.
That's so reassuring. I've doled them out. I've got the magnetic ones which I've stuck all over the lab so people can't take a picture without having one like immediately to hand. So it's great. Yeah.
And
I've converted them.
That's appreciate. That's great. And the person who did it also says they're now uploading it into Plant CV. So there's going to be within the Plant C package a kind of special system for it, which was unreal. I didn't see that coming. But anyway,
I was sharing them with people at Danforth, so getting right into a whole bunch of the plant people there too.
But anyway, just kind of riffing how like an example there is because I like to try and share like lessons learned, methods, best practice, these types of things. So that's a pun or advertising for Andrea's going to talk about a little bit later with the soil sensor that he posted in the forum, and so we'll open up in it. But I think initially we'll start off with the introductions, just giving the videos time to introduce themselves, and then we forwards. And I'm hoping the what's it called, the Algae team does turn up. So I spoke to a couple and they said they should be. Algae team, I should really call them actually, Mark and his former postdoc. But it looks like everybody is here. So I'll drop the link to the community forum in the chat, just as a quick reminder now that some folk have arrived. So we have a kind of Git book kind of notepad, we can make edits and suggestions in there. There's also within that a link to the community diary. So in the community diary, it's where we have our like evolving agenda. So quick, any ideas, anything you want to chat about, throw it into there. In addition, in that the agenda in the minutes, if you want to add your name, that would be appreciated, so we can kind of keep note as to who comes. Points mean prizes. We celebrate people that come regularly and do so in our public meetings when we come together, and that's how we can kind of keep track of that. Okay, cool. That's the kind of the housekeeping stuff. Yeah. For those who have heard it before, but for those who haven't, it's good to know that there's a structure. But is there anybody here who hasn't been to this meeting before? I'm just give people a chance to introduce themselves. We've got lots of lots of good regulars. Good to see you all. Hi. And this is I try and put someone on the spot who I don't know if you've been before or if I just haven't recognized it. Kajari Das. I'm sorry if I'm butchering your name. Have you been to this meeting before? If not, this is a chance to really introduce yourself before we go off another tangent. I've got some more people in the lobby that are just being admitted to, which is good.
Hello.
Hello, hi.
Hi, everybody. I'm Kajari Das. I actually is a biotechnologist. I worked on any type of life sciences, like starting from microbes to animal and plant also, including. So I have interest in knowing anything from this platform. And I have lots of work, published work. And can anyone go and Google search, then can see. And hope for the best. Thank you all.
Well, that's great.
From India. I'm from India, right?
Brilliant. Yeah, well, welcome. So hey, if you can come back to a future meeting, and if you like, you can open a slide deck, and you can use a couple of slides to introduce yourself and some of your research with, and kind of illustrate that a bit better to give you a chance to show what you have on offer. And today, actually, we'll talk a little bit about what the Open Science Archive has on offer for researchers like you around the world. And hopefully, so Shankir at some point is well, more than welcome to kind of take the reins and kind of present a little kind of mining that she's been doing on the OSD articles. And she's more than welcome to tell her own story if she wants. If she's in a place where she can turn on her screen. I see that Mark Settles arrived, and so he's one of the NASA's like algal experts. And Shakir came to this group a few months ago and mentioned that she's an interest in algae, and so I kind of asked her, hey, could you peek around on the website and see what you can get with little to no guidance? And then at that point, we can kind of learn from your user experience as a group, but then also find ways to help you find out more of the knowledge that's been generated by NASA about algal and algal research in microgravity and its potential niches and for future extraterrestrial agroecosystems and habitats and those types of things. So I'm saying this too much because Shankir, I'm trying to encourage you to take the reins, or if you're in a position where you're not able to, let us know. We can pivot to an alternative. But you did chat earlier. Yeah. Throw the ball, catch the ball. She's not catching the ball. Yeah, okay. So, okay, maybe Shankir is having technical difficulties at the moment and doesn't want to isn't able to take that. So, in that case, we can pivot. Okay, so Shankir, if at some point your technical issue is resolved, let us know when you can chime back in, because again, we'd like to have that conversation. But Andreas, I did notice that your screen was on earlier, and so maybe we'll pivot to the second discussion area, which was the sensor that you were developing.
Yep. Can you hear me okay? I can see that you're all frozen on the call, so I just wanted to double check if you can see me okay.
I can hear you, but my screen is personally black. I can't see you personally.
Okay, let me just rejoin. Just rejoin 1 second.
Technical challenges here. And this is why it's so much more fun in person, you know, because oh, there's another name I haven't seen before. Just he's back. Annika Sada. So not just,
I'm here. I'm here already. Yep.
Of course. Back to you, and people can try and introduce themselves a bit later. But over to you, Andreas. How's it going, mate? Like we saw your
Doing
post
well.
before.
Yes, I actually brought just 2 slides just to give a little bit of context. What are we doing, trying to do over here? What are we trying to achieve? And a small demo of the sensor, and then just gather some feedback and take it from there. So, if you will allow me to present. So, well, basically, I'm based in Coventry University in the United Kingdom. However, I'm supporting one of our labs that we have in Costa Rica. It's located close to the Carrara National Park, and basically, we're trying to introduce to our research capability the soil sensing and regolith research. So we're kind of newbies on that area. However, what is the background of the lab? And they have had 15 years of tissue cultivation. So they're especially Wilbur's, Hase, the lead in this area, in terms of replication of orchids. So they actually grab a type of plant and they replicate it under agar conditions to basically develop sustainable crops. That the basically aim of that center is to develop sustainable crops. However, our new next stage is to increase the capability and research and extent of the lab to include advanced sensing, regolith research, and as well remote sensing. One of the progress that I have done in this past year, I did a visit at International Space University in Certed in France to basically learn as well remote sensing. So along this process, basically the 2 main areas is this soil sensor that you can see over here. I don't know, you can see it on the screen. Yep. And basically what it does is that through the Bluetooth low energy protocol, allow us to capture in real time humidity, analog value. In this case is mapped from 0 to 100, and the temperature. And the reason for that is, well, because the lab doesn't have this type of equipment and we want to begin this level of research, we need instrumentation. My background is mostly on instrumentation and medical physics. So what we're trying to do is begin the ground up work, develop the capability, prepare the apprenticeship school, prepare the programmees with this new technology. So yeah, so as you can see over here, it has a quite simple. It can be powered by a Li-ion battery, which is very handy, comes very handy in terms of the kind of what we have in the lab. We don't want to rely too heavily on Wi-Fi since sometimes the network is not extremely reliable. So we want to rely only on low energy Bluetooth. So the final aim is so that we can capture all this real-time data on a local network, on a local mesh network, not only for a small range, but as well a long range for crop sustainability, and as well complement that with satellite sensing and remote sensing. And that's basically it. We just began this week since I arrived from the UK to Costa Rica. I have delivered all the materials. Bor as well, and all the team at the regolith team has been extremely supportive. So I just have to give a big thanks towards that side. We're preparing a publication. And that's basically it. That's basically it. We're actually a team of 3. Jose, which is our biotech lead, myself as more type of managing the whole project, and as well we have another point of contact in the University of Jordan, which is an expert on soils. And yeah, that's basically it. It's just quite a dream, but I think it's just going one step at a time.
That's really cool. That's really great. This for up to questions at the moment, but kind of reminds me of what is the major lesson that we often learn when we come to regolith research, and it's, well, to me, like these, and I've been reflecting on conservation. Like we can put a lot of time and effort into trying to regenerate like regolith, you know, like alien soil, or like, but conservation, like there's so many places that have this red soils deep down underneath, and we need to find ways to conserve that as much as possible. But that's just like the first thoughts that always comes to mind now. But does anyone have any thoughts or questions regarding to this? Lindsay, go for it. That's great. Appreciate it.
Hi, this looks really cool. Is it, so from what I see, it's temperature, humidity, and is it pH you measure as well?
Yes, it's actually a capacitive measure. So the sensor obtains the capacitive amount of current on the soil, and we can basically map the humidity. But based on what our experts in Jordan tell us, we can we basically get an analog reading from the sensor, and we can use it for many things. So, for example, we can calibrate it on wetlands type of environments, and then test it out and troubleshooting and prepare the sensor for different applications. Yes.
Cool. So do you think it could also be used for measuring different ions in the soil or different?
Correct.
Oh, that's so cool. Yeah. Okay.
Because
cool.
it depends on the capacity of the soil. So basically, we depart from the principle that you can make a soil battery. So if you put some wet soil and you connect them with nails and copper wires, you can basically create a 5-volt battery. That's the same principle of how the capacitive sensor reads. So you put it on the soil, and you can read the amount of electron discharge that is been present. Yep.
So does it need a certain level of moisture in the soil to function, or
no? Currently it's working on the air. Of course, we need to calibrate it. For example, if I put my hand, it will immediately detect the impedance of my hand, basically, since our hands have impedance. So that's however, the temperature remains the same because it's the temperature of my room. As you can see, it's a bit warm, and the humidity is a little bit high in Costa Rica. So yeah, that's things that we're still on. But the main objective is that it's an affordable, deployable technology for large scale applications.
By the way, you did make the right choice to go from the UK to Costa Rica this week. It's a storm outside right now.
I think the sun is setting at 4 p.m., isn't it?
Yeah, it's bad.
It's really interesting because at the moment now there's lots of different labs around the world come up with different ways to come up with bioremediative strategies to make regolith amenable for crop growth. And depending on the way you are, how you're doing it, people are using different metrics for assessing the success of their attempt to rejuvenate these regolith soils. I just dropped into the link. They're just one example, a recent paper that came out with about using algal growth to use that as initial treatment to try and make the regolith more amenable to growing crops. And so as we look at this particular paper and think about how could we compare that methodology to the other methodologies, I know that Boca is here in the room, and he's shown that you can add antioxidant treatments as one way to. So as we look at all these various different strategies that are now being accumulated by the global community, I often wonder how can we compare and contrast between them to look at the mechanisms by which they're functioning in order to work out which ones can be used additively together, maybe addressing different factors. And as I look at your work and the sense that you just described there, do you think you capture all the major key characteristics that would be needed for a lab to compare and contrast the different strategies that are being achieved? Because one of the goals we have here is to advise people like the Growing Beyond Earth program from NASA to create a standard for the citizen scientists that are doing this type of thing. And so, yeah, what does that make you think, sir?
I think our main objective, and that's a brilliant point, we depart from the idea that we didn't have any type of instrumentation. I know that, for example, from a capability perspective, Jose knows how to, for example, do genetic analysis of the plants. So he knows how to do it. And our current project is the effect of regolith simulants. We have MGS1, MGS1C, LHS1E for engineer applications, to add them to MS culture media in vitro plant morphogenesis. So that's kind of the main objective, but as well, we are open to welcome any type of hypothesis to put it on the lab and test it out as soon as trying to accelerate. I think our main objective is to accelerate and learn in the process.
I posted a link to the post that you made with the picture. Can you talk a little more about the hardware?
Of course. Yeah, so it's an open source hardware. We actually got it from China. Yeah, it's actually an embedded. Well, I can work with ESP32s. So the capacitive sensor and the soil sensing already comes with a TCB. So it's an all inbuilt PCB. The only problem of these sensors is that you have to map where the pins of the sensor. So the data, basically the data sheet is non-existent. So the process was to scan it. So we know it has an ESP32. We know it can use Bluetooth and Wi-Fi. However, we needed to know where are the sensors pin soldered. So you have to scan the board to know where they're located, and then you load the firmware. So I can just do a brief, for example, sample of how the firmware works. So basically, we have to prepare the protocol of the Bluetooth. So same as we use our AirPods or or our Apple Watches or whatever, they all use a technology called Bluetooth GET and NAT profiling. That's a service in networking. And basically, we had to develop our own AT and GET profiling to connect in a simplified way with our computers. The second step, your question, Rachel, was that we can map we load the libraries and we read the temperatures and humidity. That's already done. However, for the soil value, that's a question for that's an answer for Richard, is that we read an analog value, or Lindsay as well. We just read an analog value and we just map it from 0 to 100. Now, the work that we're doing at the moment is that we need to calibrate that value before we map it. So there's going to be some ongoing work over there. And then we just publish it. You just send the value, the values over the Bluetooth network every 1.5 seconds, because it's not a medical application. It's not a critical application. And that's basically it. Yep.
Very cool. Sorry, I just don't know anything about how the sensors work. Like the bottom part that you're holding in that picture,
Yeah.
is that what actually goes into
Exactly into the soil?
regolith? Okay, that's really cool.
Yep. And this is the one that goes.
Oh, you have it. Oh, nice.
The outsides. So this is what goes to the outside environment. Oh yes, have some questions right here. Yeah, Lindsay.
Hey Andres.
Oh, hey Borja.
Very good presentation. Very interesting. I think you have a very unique capability, especially because right now we are suggesting to try to grow plants on, so to speak, different regoliths of the moon, so to speak, right? Like we hypothesize that different regions will be better than others. So I guess my question is, since you have, so to speak, a way to measure the soil capabilities, if you could add modifications, so to speak, in vivo to the regolith, if that instrument could tell you what kind of regolith from what part of the moon is it equal to, right? So basically, like you have a capability to modify it to kind of map a different part of the moon and then grow the plants on that area. I think that's, I don't know if you thought about that or if that's something that you're looking to do, but I think it's super interesting.
Yeah, no, I mean, I think the whole objective of our center was to help other centers in the world to test ideas. So if you have an idea of any type of hypothesis, we can deploy it fast, and it will take us days or even weeks before to do like a whole protocol. So any type of hypothesis is welcome for us to adjust accordingly.
Oh, I'll wait a lengthy email from me and maybe we can set up a meeting about it, but that's so cool, man.
Yeah, yep. Basically we're trying as well to, well, it's an apprenticeship school. So it's a apprenticeship school. So many of the degrees are technical. So the whole objective is to develop high quality research on a rural area and as well, help us to develop the capacity of the future. Lindsay, I think you have a question.
Yeah, I was going to ask, if it's collecting data every like 1.5 seconds, what's the kind of storage capacity you have to have for the data you're collecting? Or is it quite like a low?
That's a great question. So the payloads are actually very small, into like kilobytes or bytes. However, we're now working with Bluetooth, which is what works. So we don't have to, for example, if we want to do like a dry run of taking measurements for 8 hours, we just pop in the dashboard, and the dashboard basically connects directly straight to the sensor. So we can, that's some of the works that we're doing. So basically, it just connects over here with the sensor, pairs up, we begin to collect the data. We can save it locally for now. In the future, this sensor can basically plug into the cloud. So we will be completely operator independent terms.
Right. And so if it's you've tried it in regolith and soil, is there a particular size of particle which it needs in order to operate? Like if you had really like large particulates, would that create a problem in measurements?
Great question. That's our next step for next week.
Okay.
Yeah.
Okay.
Because what I have tested at home is that the regolith has a lot of fine particles, so it's extremely fine. So that's going to be a problem. That could be a problem, or could be an opportunity.
I do wonder, do you need to like actually take measurements that fast. Like I've used some soil capacitance sensors in the past, and I burn them out really quickly by taking measurements every second. And then the second ones, I just pulsed every like 15 minutes, and then they lasted for years. That one lasted for years. So often we want to sequence more data is better, right? But sometimes there's reasons to sequence less take measurements less frequently. But again, if maybe you don't mind burning them out.
Yeah, send it every minute. It's not necessary to send every half a second.
But I can also see like so many applications of this because Rothamsted Research used to have like this built smart farms. What's that one down the southwest of England called? Where they built a catchment area so they could catch like the water, the hydrology cycles. They could sample at the bottom of the fields, and now they've been putting loads of like live sensors throughout fields to kind of look at the hydraulic cycle. But and it was funny because seeing that on the field scale, and then working on acid to see that in the chamber scale. So again, like that sense you've got there in the advanced plant habitat, they have layers of them. They have like a couple of them below the irrigation system, and they have a couple above the irrigation system. So you can see about how like the lack of gravity changes the water distribution, almost in three-dimensional space. But that's a tangent. Hello, Justin. How's it going, mate? Go for it.
Oh, I was gonna just ask, why not do this with like a more open standard, like a Raspberry Pi or Arduino for the hardware instead of the Chinese thing that you said you had to scan for the sensor pins and everything?
Oh, great question. Yeah, and the thing is, it already comes. Everything is solder now, so we don't have to solder everything to the ESP32, and the ESP32 can communicate with Arduino. So how we set up the drivers is through Arduino. Justin, over here in platform, we can basically set up the framework of how we're going to build it. So we can basically program through Arduino, and we use some Arduino libraries as well.
Oh, so basically all the sensors and everything were already part of the device instead of having to connect them.
Correct. I think it's a good next step as a next step to develop our own sensing. However, that takes some. We have to do the PCB. We have to set it to coast analysis and so on.
Okay, I worked on another project where we had Raspberry Pis and we would solder like a tongue depressor type stick, or all the sensors onto that, and you could just stick that in the soil, and then they would connect through to the Pi. But you're right, that took a lot of soldering and extra work to get all those sensors built. So, okay, sorry, I misunderstood where.
No worries.
Which part was the brain of the thing or whatever. Thank you.
That's right.
I'm also thinking about have you normalized like the depth that you put it? I played some sensors, and depending on the depth, I got different results. Like just have a system for standardizing that or controlling that.
Excellent question. Good action item as well.
Okay, cool. Okay, so now the space to aspects of it is yeah, how would you prepare this for flight? Okay, so you're producing proof of principle data on the ground, right? Showing how this could be useful for assessing regolith in situ, to help guide potential augmentation strategies to make it usable. But based on your use of the system so far, how would you adapt it? How would you
That's an excellent question. I think one in the forum, I think a scientist wrote that you can prepare those sensors for space. I don't really understand very well how you can prepare. I think there must be a type of coding for the radiation, but I welcome any kind of suggestions on how to send that if the future.
There is one that I've become aware of. If you have any rubber components on the system, there's different ways. If you have any rubber components, I didn't see any on yours, but one way out that people
only I think that the blue one is just plastic, the cover plastic, so there's no rubber.
And so one way people prepare rubber for flight is actually removing all the oxygen from it, because rubber kind of conducts like air through it. It's able to because it's kind of nature. And so if you can make sure that there's no oxygen in the rubber, then it will last longer in microgravity in flight. So yeah, so degassing plastics and related oxygen is one mechanism for which people provide resistance to some of the effects of galactic cosmic rays and gamma radiation in LEO. I only found out about that one recently, so it's but yeah. And I don't know how about the shaking effect. So the shaking table is the other thing there's the other major challenge you always have to get past to try and get something to fly. I don't know if the NASA people can add more detail than the shaker table. But there's a certain like vibrational load that all kind of flight hardware has to get past. And then it goes through like the technology readiness level. So even if you've done those, you then have to get it like codified bureaucratically, so that people know it's gone through these tests. And then the next step on that is trying to give it some sort of flight pedigree. And so parabolic flights and the next kind of sort of intermediate people often go to try and show that their hardware is capable of withstanding a launch to LEO and beyond. It's elementary, really, but again,
plenty of work, I bet. Yeah, I definitely will give a read to this and have more questions for you.
And it's one of those requirements that's the same in all space agencies. Even the Brazilian space agencies have the same kind of shaking requirements that we do. Hey, Mark, how's it going? Over to you, sir.
Yeah, just a comment. There are a series of different, it depends on the mission class, whether or not you're going into a human-rated spacecraft, other things like that in terms of like what are the exact requirements for the hardware to be able to withstand. So like for Eclipse mission, you have more of a do no harm to the rest of the payloads that are on a non-crewed, like an autonomous lander. But for human-rated spacecraft, there's a whole bunch of safety issues in terms of volatiles and other things like that, and hazardous materials, flammable materials. Almost everything is flammable. So it kind of depends on where you're planning on trying to take the hardware, right? Where it's going to go. Yeah. So if it's going to go like in a CubeSat or something like that, that's much. That has lower concerns about human safety, but it has different concerns in terms of being able to continue to operate in extreme environments. Yeah.
Okay.
So I'm thinking now of like Ohala, like the new piece of spaceflight hardware that's just coming along. So I say this because the current or most recent state of the art of spaceflight agricultural research hardware has been the Advanced Plant Habitat. And in that system, Sierra Nevada Corp built into the science carriers, and that's like the tray that had the arsenite, which is a clay material from Earth, which is kind of similar in some ways to regolith, right? And in that, they decided to have multiple conducting electrodes, like the ones that you're using for monitoring the water distribution. That piece of hardware has now been discontinued. So the next piece of hardware that's now going to fill that niche and is bigger and is better, apparently, essentially, is the Ohala. And for that, again, has some new science carriers that are going in the base of it, slightly different configurations. We'll probably have some kind of lessons learned from the prior astroculture methodology that was used, but will be slightly improved and better. So as I say all this, I'm saying because we know that there is active development in that area for new sensors, enhanced sensors there. So that is like an opportunity that always kind of we want to encourage and discuss with people. And that reminds me of that paper that came from the Kennedy Space Center where they had their knowledge gaps that they identified for like crops, fundamental biology, and the others. So I'll dig that paper out and kind of drop it into the chat here. So just kind of say hit into along the lines that we know that the types of sensors are now under review, and it makes me wonder like how to help compare your sensors to the other state of the art to try and work out what needs to be done to move the field forwards. So that we do have sensors that won't respond to that, say, to cosmic radiation. What does that make you think? Okay. So it's definitely not a lot, didn't you? But
I think that's very ambitious. I think our main aim was to develop our like local capability to have the instrumentation, but I think having I've always wanted to develop space technology. But I think all of these small details are very important, because there are many things that we can begin to implement now, at least from a PCB perspective. Like how can you not have a PCB ready for spaceflight, but at least have it for a high degree of engineering. So it has a high resistance, it is reliable, it is, and I mean, at least can not withstand space, but at least withstand extreme environments and extreme humidity. So I'll definitely have a ton of questions, like tons of questions, because it's a whole new area for me, is preparing something like that.
And again, and that then makes perfect sense what Mark was saying, how in space biology, there's a ton of different destinations and goals. And so you came here focused in on regolith and applying the technology in remote environments, tropical environments, back where the conservation message is so important. And we were just then highlighting how with all the vast array of different places that humans need to thrive in the future for us to go beyond a low Earth orbit, there's different ways that your sensor as a baseline can be adapted and hardened to make it appropriate for those different like mission portfolios. Like again, like Mark was alluding to, each mission's portfolio profile is slightly different and slightly different requirements, right? Okay, cool. So, but in order to trying to find the OSDR link here. Hey guys, whoever's rejigging the OSDR website, it's way harder to find stuff now. Like I know that GeneLab, like
Richard, it is not within our control.
I know.
But thank you as always for the feedback.
Yeah, you know me in the best possible place, and I know it's not you. But you had it better previously. Is what I'm trying to I guess. But what I was trying to say is I'm trying to find the Open Science Archive, because essentially one way to show that this technology can help move the field forwards is by looking at the prior art and trying to see what studies have been done by NASA and NASA-funded researchers in the space biology community globally, and try and see what we can mine from there to help move us forwards, as it were. The thing I was going to then do was do an algal mine, because that's something we discussed previously. But I can't find the repository page because someone's been juggling the website. Oh, Nick, say it, don't type it.
Hey, I'd written a really nice, meaningful message.
Hey, Richard, just one, real quick. Do you know the AWG about page? Are you familiar with that link? We do have control of this link 100%. So what I've done is, yeah, you're not the first person to point out that OSDR is almost unusable now because it's hard to find everything. So, but the about page for the AWG forum space, yeah, the about page, not just the home page, but the about page, all the one I put in there, all quick links are 100% accurate 24/7. The tutorials, the repo itself, you know, the Rad Lab, Splash, the educationals, the data standards, the Google Scholar, blah, 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 blah. So everything is up to date in there.
That's better.
Yeah, so I've been using that as like a central place now to get all quick links. Yeah, because like Chris said, like it's the NASA website itself is out of our hands. It's way over our head. So, but the forum space is in our control. So that's why I made all quick links perfect in there. And if anyone has other links you want me to add in that about page, please let me know.
Yeah, totally. Because you used to be able to just Google OSDR repository and it would just come up, and now it takes you to all these other websites before it, which is all this new NASA front end, which looks very beautiful, of course. We love it.
I sent a link that I think is filtered by the algal studies, but I'm not sure. I sent an email to Shankia, and she said that she can hear us, but I'm not sure. Are you there?
Yeah, I come.
Yeah, I'm not sure.
Because this is her chance. We did try.
It's weird because she's listed as an attendee, but not a presenter on the participants. So there must be something going on.
Yeah. So in the meantime, Mark, what was that wonderful thing that you presented at the STAR meeting that Lindsay mentioned?
So I give a talk on microbes in space, basically, like how at least we're doing stuff at Ames in my research program. So it's like about a 45-minute talk. Yeah, but like if people, I saw, I was just about to, right, Rachel, I saw that you were asking for you were planning a short talk on algae. Was that somebody else, or were you actually hoping I would talk about it?
No, there's someone else that was supposed to give a short
Okay.
presentation, but yeah, I'm not sure what's happening.
Yeah, the
Okay.
communication's not working at the moment, unfortunately.
Okay.
But appreciate you able to come and yeah, kind of provide feedback on that. But but you said microbes. I always thought your research was focused more on algae. Like, what's your interest in microbes? Can I mind you?
So I do, yeah, I work in synthetic biology. So actually, maybe I'll come on camera. It's a little awkward. It's my webcam doesn't work anymore, so I have to use my laptop. So yeah, so I work on synthetic biology, both for yeast as well as for trying to look at algae and even plants, right? So so I'm the PI of the Leia project, which is going to send yeast that are engineered to synthesize beta carotene to the surface of the moon in the South Pole region. And with that particular experiment, we're trying to look at whether or not deep space environments would wind up giving us a yield penalty. And that's kind of important. It would potentially lead out to thinking about algae as well as other crop organisms like plants and things like that as well. And so a little bit, it's a kind of a different direction than what LEAF is doing, because we're very much looking at very quantitative metrics of growth using spectroscopy-based methods of detection. Yeah. So yeah. So that's most of my funding is actually in working with microbes, working with yeast.
So what trends would you hypothesize you might see in the lunar gravity environment? And what trends might you hypothesize you see because of the radiation aspect? Just
yeah, so like what we see on Earth with yeast that are engineered to produce beta carotene is that they are very sensitive to abiotic stresses in terms of their yield, but not in terms of their growth. And so you can get good growth, but then you'll get less carotenoids, say, in a high temperature environment. And so in a high radiation environment, there's also the potential for that, but it's fairly difficult to give the mixed field types. It's actually impossible to give the mixed field radiation that yeast would experience on the surface of the moon, where you have a mix of galactic cosmic radiation, solar particle radiation, like solar wind, and then the albedo, where you have both fast neutrons and gamma radiation coming off the surface of the moon. And so that may be one of the things that we would see is potentially lower beta carotene production, even though the yeast will probably grow around the same rate as what we would see on Earth in our ground control. So that's one hypothesis. The other one is that because there's partial G, that the yeast may not settle as quickly. There may be the ability to have a little bit more suspension of the yeast, so they might actually then grow a little faster because of that. But we don't know, right? Like they should still settle, and so to the bottom of the culture bowl as they grow. Yeah, Bora.
Hey, Mark, quick question. So compared with other organisms, what would you say the mutagenesis rate is of yeast in deep space radiation? Do you think, obviously because the yeast will be growing and growing, but like I wonder what is the mutagenesis going to be like in the early, so to speak, colonies, if you wish?
Yeah, so we've I don't know, were you at ASGSR?
This year, no, sadly no.
Yeah.
I miss all of it, and I miss all of you.
Oh, yes, it was a difficult year. So Philip Sweet from my group gave a talk on looking at the bionutrients experiments in low Earth orbit. So that's kind of the only data that we have in terms of mutagenesis of stored yeast. And so there, the experiment did store the yeast for long term on the ISS, and we saw a very low, as long as the yeast were desiccated, we saw a very low mutagenesis rate, and we saw a very long viability for the yeast. So they were viable out till the entire experiment, which was 5 years of storage on the ISS. And so my guess will be that we won't see, the most of the mutagenesis that we actually did see was more correlated with loss of viability. And so then you're kind of going, well, is it the abiotic stress of just sitting there desiccated and degrading over time, or is there actually really much of a impact on organisms that are essentially in stasis in space? And the same thing's been shown with tomato seeds, right? Like in the 80s, there was a NASA mission that held tomato seeds in the vacuum space for like most of a year. And when they came back, all the seeds were viable, and the mutagenesis was not that high. Like you didn't see large amounts. Yeah, go ahead, Richard.
So then what would you expect? Sorry.
And Richard also had something afterwards, I think.
Oh,
Yeah.
sorry. I was just curious, what would you expect to see in algae then?
So yeah, actually, I mean, if you want, I can show like what I have done as a chlamydomonas Reinhardtii experiment that I could give like a short presentation if I find it. It's from a long time ago, but we just recently.
And I just, as you load that up, I'd intervene and say, well, it depends on the oxygen, how much air is there, right? They can really dramatically affect the response. Potentially, that tomatosphere, they were pretty well kind of vacuum sealed up. The other thing, it depends on the state. And so as you're saying yeast here, I'm not thinking, does it form like a spore state? Because I know that
this was actually, yeah, the long-term storage, we had both spores and one version that was with spores and one version that was just dehydrated, desiccated, and saw very similar phenotypes, very similar genomic responses between the two yeast strains. Yeah.
I'm like, I'm really excited. There's this new paper is just coming out from when they had moss spores on the side of the space station, and they did like the expose on the outside the Kibo module. And so it's like plus minus UV shielding, but essentially they're able to show that they were able to survive for like 9 months up there. So I had this image of like Starship flying to Mars and all these like moss spores like hitchhiking, like stuck on the outside and going over there and being like early pioneer terraformers. But I've just gone off on a sci-fi tangent to fill some space. But the new paper is out on the Physical Shadow out in space, and looks quite interesting. At least it should be out soon. But seeds.
Yeah. So would you like me to kind of talk about that spaceflight experiment? I have like a presentation that I gave in Plant and Animal Genome a couple of years back. If you'd like to hear about it.
Yeah, that would be fascinating.
Sure. And it's a little bit out of date for like our current Space Algae 2 experiment, but this one is, yeah. This one is available for open use. Did it play? Come on. There we go. Let me get you guys small. Okay. Yeah. So we've got a couple of different projects. One that was occurred before I joined NASA in 2018, which is the Space Algae 1 experiment, and that was funded by the Florida Space Grant Consortium through the University of Florida and University of South Florida. I'm gonna grab a pointer. And so like the big thing of like what our motivation for that first experiment that we've actually got data from space about was basically thinking about how would you go from everybody proposing algae as something that you can use in space to actually being able to grow algae in ponds or grow algae in mass for real bioproduction in deep space. And so one of the big thoughts about that is that potentially growing algae, growing anything in space would likely require some genetic improvement in order to be able to adapt and grow in whatever environment we're actually going to grow in. And I mean, we saw some data at ASGSR talking about how much rodents react to just the habitat that they're in, and that the space itself, the space flight part, is not actually as big of a stress or as big of an impact on say your genomic responses as the growth chamber itself of what you're growing in. And so the adaptations that we need to make to either grow in microgravity or to grow on the moon, it might be the biggest problems might be actually adapting our crops to those growth chambers so that they can grow very efficiently. And so my one thought that we had was, one motivation for this first algae experiment was to see whether or not we could do a competitive growth assay to try to understand what genes are important for algae to be able to grow in a spaceflight environment, more specifically in a particular kind of hardware that might be applicable to grow algae in long term. And so what we wound up doing in Space Algae 1 is we took Chlamydomonas reinhardtii, which is both a model organism, but now also a grass, or generally regarded as safe organism for consumption. And we pre-mutagenized it 5 days prior to starting a mixotrophic growth in veggie. And so by mixotrophic growth, what I mean is Chlamydomonas reinhardtii can use acetate as a carbon source. And so we included that into the media in part so that the algae would grow fast enough that we could complete this experiment in one SpaceX mission. So like the CRS-15 resupply mission, so that the experiment would start at the beginning of the mission, and then they would wrap it up and bring all of our samples home at the end of that 35-day mission. So the challenge, of course, the challenge at that time for me was like, I didn't know whether or not mixotrophic growth would be something that would be of interest to NASA, but only later, when I arrived at NASA, did I realize that there are a series of physiochemical methods to take CO2 and turn it into small carbon molecules like formic acid and acetate. And so this is actually a good model of like how you might boost the growth of organisms. So like Chlamydomonas being a acetate feeder could potentially have its growth boosted by that. And so the basic idea is like, okay, you mutagenize haploid cells beforehand, and you get some variants that are in there. And then as you go through rounds of growth, the population, if you sample a small subset, you will wind up eventually shifting the population towards cells that grow faster and get rid of cells that aren't happy. And so, and we also did this with 2 different strains of algae and 3 biological replicates. So another big challenge of spaceflight is always having hardware that's very simple and easy to use. And so like for us, our payload was simply a foam cutout box that had a series of FEP plastic tissue culture bags. And you can see the mutagenized cells, the 3 replicates of 2 strains right over in here. And it was launched in 2018 and got installed in veggie. And then we did a 52-hour delay ground control in part so that we could, so that we didn't have to do things in the middle of the night primarily, but also we used our backup mutagenesis in case the launch had been delayed more than 2 days. Okay, and so then what happened in terms of the growth is it was grown in veggie under relatively low light conditions, more appropriate for Chlamydomonas. And then with each cycle, the astronaut would come, set up and track our bags based on color and tags with colors and numbers. So the color indicated the mutagenic lineage, and then the number was the passage or the culture number. And so they would come in, take half a mil of culture and put it into the next bag. And then some safety issues, like we needed a stopcock so that there wouldn't be any leakage of media or culture. And then to return the samples, we had done pre-flight experiments where we found that we could store these cultures fully grown like this back in that same cutout foam bag or foam box and return them, and we would still get good enough DNA to be able to do whole genome sequencing at the end of the experiment. So the big thing that was different between spaceflight and ground control is that this is a black and white image looking down. So like if you go here, looking down onto these bags, and this is from our spaceflight, and this is from our ground control after the end of a cycle. And what you can see here is in the spaceflight, the cells are very well suspended, while on ground control, all the cells pulled onto the bottom. So this is kind of like a sheet of cells that are on the bottom of the bag. And we actually did see, like, in our ground control, that biomass was kind of stable and consistent, while we saw increasing biomass in spaceflight. Part of that, though, is remember that this might be representative of the actual biomass in spaceflight, because those cells might have been dying and falling apart because they were not seeing any convective mixing for permeation and diffusion of gases across the bag. So this bag is actually, it allows some CO2 and O2 to cross it. Okay, so then when we did our whole genome sequencing, we then used a pipeline to identify single nucleotide variants in both spaceflight and ground control. And when you look at this, you can see that, if you look carefully, you can see like the density of mutations might be a little bit higher in our ISS samples, which are in orange and blue. Gator colors, right? And then when we actually looked at the individual point mutations and what sites were changing, we actually had more mutations in our spaceflight strains with about 10,000 more, 10 to 11,000 more total point mutations identified in the spaceflight compared to the ground controls. And looking at the relative contributions of these, the one thing we noticed right away was that there was a lot more T to A mutations in spaceflight. And when we think about the radiation exposure there, that was a pretty low exposure. So only 285 micrograys per day based on data I had back a while back. I think OSDR gives a slightly different level, but it's pretty close to the 285 microgray per day. And so this is like basically a fairly low dose of radiation, but we're seeing a fairly high level, a surprisingly noticeable and significant level of additional mutations in spaceflight. And, just to give you the contrast, so like these guys have C to G mutations, which are one of the signatures that we would see from UV, were very similar between spaceflight and ground. But then when you look at T to A, you can really see that they're distributed all over the genome and at much higher density than what you would see in the ground control. So, that suggests that that in this particular case, this particular organism has some sensitivity. And when we now look at in 3-base context of these, those T to A's are in a GTA 3-base context. And during the shutdown, I was actually able to kind of look into more detail about what that signature or that particular context means. It happens to correspond to where methylated adenines are in the chlamydomonas genome. And if we look at compared to say human profiles for 3-base context mutational profiles, what we see in the ISS is a series of signatures of mutation that are associated with a couple of different things. Some of them as being radiation, and some of them being different kinds of toxins that are used for cancer therapy. Then what we saw in ground controls and in ISS and ground controls are things that would be related more to UV damage repair and DNA damage repair. And I've actually got this updated, and these are not exactly the etiologies that we see. They've been changed a little bit in the time since I made this slide. And then we also looked at selection, looking at the rate of synonymous to non-synonymous mutations inside the genomes and inside the genes that were mutated in both space and ground. And one of the some of the core things that we identified was DNA repair mutations were selected, for in both space and ground, and that makes sense because we did a pretty hefty mutagenesis. And then there were also some of these like ATPase and zinc ion and transition metal binding and things like that were under selection as well. But those some of those turned out to be purifying selection or positive selection on factors involved in chlorophyll. And in fact, one example of this is the NAB1 gene. And so that is an RNA binding protein that, and this is coming from other papers, an RNA binding protein that when mutated increases the amount of chlorophyll content inside cells. And then the CHLI1 is another one that was involved in chlorophyll biosynthesis. And when that is mutated, it basically knocks out the ability of the cell to produce chlorophyll. And so both of these had, so the spaceflight samples had mutations in NAB1 that were in both of the conserved RNA binding domains of NAB1, so they might be reduced function mutations. And then we also saw mutations in aspartate to asparagine in the C-terminus of CHLI1. But whether or not it has a loss of function or gain of function phenotype is unclear. So we don't really know. And so coming out of space algae 1, it's like is ionizing radiation that we see in low Earth orbit really that bad, or is it just something kind of specific to this particular organism? And then if there is mutation occurring in space, like if you were planning on producing algae for a long period of time and basically sourdoughing it, propagating it from one batch to another, or doing a continuous culture system, how much risk of genetic drift or selection might there be? And so what we were then interested in doing and wrote a follow-up grant for Space Algae 2 was to try to look at a different organism that's been much more studied for space research and for space bioregenerative life support systems. And that's to look at Arthrospira platensis, now Limnospira platensis, but commonly known as spirulina. And so that particular cyanobacteria has a very high photosynthetic activity, efficiency. It's also a good protein source that's been used, was independently discovered both in Central Africa and in Mesoamerica as something to make these protein cakes out of. And the Melissa Project has been studying this for decades to see whether or not it would have good photosynthetic efficiency in space. And in fact, they had a spaceflight experiment called Arthrospira B, where they did show that spirulina has very good photosynthetic parameters in space. The other big thing that for us, in terms of thinking about genome sequencing, is that it has a very small genome because it's a bacteria. And so that allows us to do a lot deeper sequencing with the same cost as what we did with Chlamydomonas ringhardi. And so what we have done over the last couple of years is basically develop all of the pre-flight testing to get ready to send this to space. What we're planning on doing is basically growing the spirulina over 5 to 6 months, and then passaging it basically every 2 weeks. So we've worked out conditions to slow the growth from about 6 days per cycle to about 2 weeks per cycle, and then purifying out or dewatering the biomass for freezing it so that we could then do genomics, transcriptomics, proteomics, metabolomics, right? With Space Algae 1, all we could do was whole genome sequencing because those cells had been sitting in a culture stagnant for up to like 3 weeks before we did any work with them. And so the basic idea is that every couple of days or every couple of weeks, the astronauts will come and do a transfer, and then those old bags would have, at the same time as the transfer, passage them back. And so this is just some examples of some early data. We actually have more data now that we've got a pretty good concept of operations. And I think that's basically it on this little presentation. So this is from our last cycle of growth. The algae were grown in this veggie unit over here. You can see it's very highly red. And this was when they took the last cultures out, they were kind enough, the astronaut was kind enough to give us a microgravity spinning bag of algae as a video. It was really cool. Okay, I think that's everything for that. I'll stop sharing. Yeah.
Love it for the spinning on. Seeing that spinning bag, can you ask them to spin it for enough for 1G control? Like...
Yeah, it would have been funny, right? Yeah.
Oh dear. Oh no, that is awesome. Do you have any questions regarding? Mine are kind of trivial, just like looking at the different like strains and colors of spirulina. Just wondering if you ever assess the different colors to see if they have a different effect on their responses to. Because there's I'm reading around. Looks like there's a blue variety. I've seen some red algal varieties. What does that make you think?
Yeah, so we have kind of focused in on one strain that does have some spaceflight heritage. There's actually a second strain, which is the one that is the focus that the Melissa Project has more focused on for their spaceflight work. But in part, we've focused on heritage strains mainly so that we could build on the prior work. However, the ones that have really different colors, and some of them are actually different species, but like ones that produce astaxanthin, there are microalgae that produce astaxanthin, which is thought to be a very powerful antioxidant. And so if you're going to take a supplement, that might be a good one to take. The only problem is if I remember that particular species, you really need to extract the astaxanthin from it rather than just directly eat. And so part of the other part of our rationale for focusing on spirulina, on this particular species of spirulina, is that this is a species that you could dewater and directly eat the algae without doing any processing whatsoever. Yeah, and you can even screen for for high levels of contaminating microbes by just in those growth bags. You can put them onto a microscope and very easily see the culture and see what's in there. Yeah.
Katie told me that when she started working on this, she started putting a scoop of spirulina in her smoothie every morning.
Yeah. 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 I mean, it does have it's a complete protein source. It's got nice vitamin content, antioxidant content, all those kinds of things. The only thing is that it does have a very fishy taste, and it's exaggerated by drying that taste. So probably better to eat it fresh than it is to dry it out and try to take the powder. The other thing, Richard, I looked at the paper that you had linked through. It's kind of interesting how they didn't really describe the strain or how they produced the spirulina, and then when they made their solutions, there was like by weight or by mass, but not clear whether or not that was like mass of culture or mass of cells, fresh mass, dry weight. So I think that one would be really hard to repeat the spirulina supplementation without going to the authors. Yeah.
That's an interesting point. Yeah, I've noticed a couple of gaps in it too, but and I guess that's the importance of the OSDR and trying to, because sometimes you don't know you're missing an important piece of like metadata or methodology until you speak to a subject matter expert, and they're like, oh, actually, it's not reproducible unless you tell us how you grew it initially or whatever.
Yeah, so but like if anybody's thinking about trying to do that kind of a replicate study of something like that, yeah, it would be those would be key questions to write the author about, right? Like how exactly did you make this supplement, this fertilizer? Yeah.
What is like a standard bioreactor for algae? So the ones that you selected for flight had to be selected because of certain flight pedigree, those types of downside things.
So for Chlamydia, the space algae one, I don't think we did not have any good examples of anybody growing algae in liquid culture. So everybody who had grown algae prior was growing it on solid media. But the growth rates are so slow on that you couldn't actually use it for production. And also because they're physically where they, in the selection pressures on that wouldn't be very accurate if you were trying to select for production. So yeah, I looked around a bit and tried to figure out, okay, what are the containers that we could use to grow algae in liquid, and wound up thinking, looking at bags that, like, for transfusions, there are certain blood bags that are needed to keep cells alive. They have a particular kind of plastic. And then I also found these tissue culture bags. And then only after I started did I realize that NASA knew all about this. They use FEP bags all the time for everything biology. And so the features of that particular plastic is that it's very resistant to water permeation, but it still allows a lower level of CO2 and O2 and other gas transfer. The one gas that really doesn't go through is nitrogen. It gets trapped by the FEP for some reason. And so in terms of like as a bioreactor for liquid cultures, I mean, that's been repeated since then. I think there was another algae that did an astaxanthin production system, but it only put up on the wall instead of put into veggie. But the other bioreactors like Melissa project, and then there's also a German group that has produced a capillary, or like a not capillary, like a raceway plate for algae that has half of it is FEP, the other half is a solid support for doing a continuous culture. But yeah, there isn't a standard for algal bioreactors. We're just going with the FEP because it's simple to implement. Yeah. So
I'd admire Flask on a shaking incubator in a control chamber. Is that like sound?
That's what we tend to do as our reference. Like what would be the optimal growth rate. And then the other thing is that big factors, just like with yeast, temperature, light levels, the amount of light levels when you start your growth of your culture should be lower until the cells get enough shading of each other so that they're not getting photo bleached and things like that. Yeah.
I've talked to someone, you or Katie, about this before, but I just wanted to bring up, so if you were going to use this as a nutrition source and grow it very long term, very far away where we have limited numbers of bags, what would you grow it in?
Well, I mean, you can reuse those bags. Those are completely reusable. What would I grow it in beyond that? I mean, there would be, you would probably want to have a more continuous culture system at that point to develop. So you would need like real hardware, and you would at that point you would have some flexibility. So it's a question of like, how are you going to do your gas mixing? Is it going to be a microgravity system, or is it going to be a partial G system? If it's partial G, you can basically use systems that people produce algae on Earth with. But if it's in microgravity, that's where it gets complicated. Yeah. And microgravity bioreactors is still, that's for microbes is still a challenge, because gas mixing is really problematic, right? The phases and getting them to really agitate and exchange, because it's not like, for plants you can at least put fans and move the air over the plants, right? Yeah, but I don't know if you've seen like how bubbles in microgravity behave, like with the astronauts and stuff. Yeah. Yeah.
Yeah, those are always fun to watch when they.
But they really illustrate how it's diffusion limited, right? And that it's not actually, the bubble doesn't go up, it doesn't go down, it doesn't go anywhere. And you have to actively mix it around to get the gas to exchange with the liquid.
And that's one of the things I found most fascinating, because I met this company called Algae when I was at Rothamsted Research. They had one of their CEOs come to visit, and they were putting transgenes into algae, clues in the name, to try and make like different types of oil. Some of them, like, they wanted to get into the biofuel areas, so trying to make them longer and less unsaturated, which was different from the nutritional team, who was going off on a different tangent. But because they wanted to get into the oil niche, essentially, and they were concerned about the algae escaping into the wild with the transgenes, because once it's out in the sea, it's out. So they realized is they could make it. They put a mutation in that made an obligate high CO2 troph. Not the correct sentence. They made it so it had to have extra CO2 being pumped into the water in order to maintain its growth. So then that way, if it escaped into the wild, it couldn't get that level it needed. And so they were able to do this because they were based in Israel, so they were near some hydrocarbon sources. And so essentially that also made the whole thing grow much faster. And their goal was to try and scale this up next to traditional sources of hydrocarbons, thinking that the traditional oil infrastructure could also be used to distribute it in the long run. And so that way you're not directly competing against the oil industry as a replacement. You're saying it's like something that has to go into that system. So it was quite an interesting way of both developing like the trans angle approach, both protecting the wild from its potential escape, while also kind of engaging with the industry that's most likely to like shut it down. But all of that just made me kind of appreciate how difficult it is to keep strains alive. So when you have a good algal strain, can you just throw it in the -80 is in a glycerol stock? Or
so Chlamydomonas you can, and that was one other thing that we had to work out for Arthrospira platensis, right? So we did actually screen through a whole bunch of cryoprotectant agents and figured out, and there's a bioarchive paper out on our cryoprotective method, which is basically a combination of trehalose and DMSO instead of glycerol.
Yeah. I should write that down, give it a try. You can publish it, I'll find it. When you put
if you just look like cryopreservation of Arthrospira platensis, I can probably grab it real quick.
But so when you brought it back, did you use a fixative with your algal 1 mission, or did it just just into -80 essentially? And was that to just freeze it? Because that's not always an option. I'm just wondering if that was a chemical step or no.
Yeah, so for the Algae 1, we in order to fly quickly, we had to we had to and to get through safety, it was like multiple things that were problematic. We had to get through safety, we had to reduce our crew time. We needed to make sure that it was only one cycle of 1 month and so on, because CASIS and the Florida Space Research Initiative, like those two groups were like, no, you have to be very efficient because this is a small grant, and so we had to make some compromises, right? And that was one, not being able to cryopreserve the algae for the return.
So it came down at room temperature, or came down alive?
Room temperature, yeah, just straight to us, and we picked it up in California because that's where the SpaceX capsules were coming back at that time.
Cool.
Yeah.
So do you still have some alive growing? Do you still have those strains? Like,
yeah, well, we propagated them for a little while, and then I should have frozen them, but I did not. I should have made stocks of them all, but we just didn't. There were many other things going on in the lab at the time, and so it was just like an oversight that I should have. I regret not having done. Yeah,
we'll keep bringing that up every time we meet. Then poking this. I'm only kidding.
Yeah, I still have a little tiny bit of biomass, like if we got like if needed to reanalyze something or to confirm something. Yeah. So, but that's another, like you're familiar, Richard, right? It's like it's really easy to have your the spaceflight materials you get back is very, very small. Right?
And it's not usually until 2 years later you realize what the thing that you really wanted was. Like our friend Jacob Torres at SGSR mentioned that I had a similar question. Oh, what about the chilies? Did you not get a seed? And he had that same kind of thing where it was like, oh, in hindsight, there was a moment when we could have got the seed, but because we didn't ask for it 2 years earlier, we just didn't,
You didn't get it. Yeah.
Yeah.
Yeah, exactly. And it's like, the person that I had sent a postdoc to work on it, and the postdoc, we had very clear instructions of what the procedure was going to be, and he just didn't follow it. So it was just like, okay, I got what I got. So sometimes it's dependent upon everybody doing exactly what we all agreed on, and that can happen as well. So
good lessons learned there. I do love those. That's the most important.
Yeah. Yeah, you should have. So the way that we deal with this in Ames is everything to do with spaceflight has you actually have the technical engineer or the technical scientist who's doing the work, but you also have a quality control scientist who's right next to them, making sure that they follow the protocol and do the procedures, and you take the notes for them so that so that if they do any deviations, you know exactly what was deviated from, and you keep that in an electronic notebook. And so if you're planning any of those types of experiments, for your lab and you actually have a spaceflight experiment, please do that. Right, have a quality control person while the critical experiments are going on. Yeah, and part of the problem that we couldn't do that is we were so short-staffed that like I had to be around to deal with the ground control while the postdoc was off in LA, in California. So
many moving parts.
Yeah, that's the other problem. Yeah, and if you can, many people like being involved in spaceflight, so you might be able to just get a volunteer for some of those critical time periods to get your quality control in. Yeah.
Well, Rachel, go for it.
I have another question, sorry. You mentioned the change in photosynthetic capacity, and obviously algae is very important for producing oxygen and could be used for space. It would be interesting to see experiments where you can get like automatic readings for that. And I don't know if there's a way you can do it I don't know if you could do it in the bags, but that would be cool to see.
Yeah, you can have oxygen sensors, and like so in Space Algae 1, I don't think we produced any oxygen because the mixotrophic growth, right? But in Space Algae 2, those bags do produce oxygen, and the oxygen concentration will go up to about 30 to 35% in the gas that's inside the bag. So
yeah. So for that, you have to be forcing the air over the surface of the bags with forced convective mixing, right? Because it needs like a wind speed of like 2 meters per second or something for exchange to occur. Otherwise, we won't get up to like.
So the exchange is slow enough that through the bag that we don't worry about it on the ground. We just allow convective mixing, but we might be able to get better exchange if we more actively moved air over the bags. But in space, we do need air moving over the bags. Yeah, because otherwise you'll get a pool of oxygen depleted air, or you'll get a pool of CO2 depleted air around the bag, and it'll just become a very concentrated oxygen bubble. Not concentrated, but above normal.
I was referring to the fan because one time I put some in a chamber, and the chamber didn't have a fan moving it around, and then like just didn't see the growth. And then when we put the forced convection in there, then suddenly there was a growth rate. We can detect oxygen production and stuff. So
was that in space, Richard?
So that was on Earth. So a bad incubator on Earth with poor airflow creates phenotypes that's really similar to flight.
There you go.
Yeah, and then when you're trying to show that you can grow something well on Earth, it's not good to accidentally forget the airflow. In hindsight. Any other questions? Oh, we've got Mark here. We've got like 5 more minutes officially on the schedule. Again, if people don't have any questions, we can always go have a bio break early. But this has been really, really informative. So that was all based on your previous mission. There's one more mission coming, is that right?
Yeah, so where we're at right now is Space Biology needs to contract the implementation partner, and I think they've gotten through the bid process, and they're just like, it's like execute the contract. And it's been there for a while.
Will that be on the ISS?
It should be on the ISS, yes. Yeah.
So my question is, from this, have you learned what's required to grow algal efficiently so that it can be used in some kind of, let's just say, an algal food computer, condensed into a system like that could be deployed on each of the private commercial LEO destinations, to provide certain ecosystem niches, or what more things need to overcome to us to achieve that goal?
Yeah, so you would probably want to have more automation than what we currently do, right? In terms of like the adding new media and removing biomass. But in terms of the core components, it's very similar to what you have on Earth, right? Like you just need media with the appropriate salts and inputs and you know, for example, like spirulina produces everything it needs itself. All it's really needing is just core salts that have the nitrogen, potassium, phosphate, and a little bit of sodium, and then all of your trace minerals that most plants would need. And so and yeah, that kind of nutrient solution and then low levels of lighting, right? Like 20 micromole to 40 micromole par is all you really need to grow the algae efficiently. The other big one is like if you're going to go with that low level of light, you would probably also need to have systems where you have good gas exchange and also good light exposure.
That's the hard part. Like it seems plausible with when you can bubble the air through it. Like then I can kind of see it and kind of start to put the jigsaw puzzle together. But as soon as you remove the capacity to like bubble the air through to kind of push in those gases into the water, it just seems difficult, but not impossible.
Yeah.
Right?
So and we've looked at a fair amount in thinking about yeast and bacterial recombinant protein or other carotenoid type production, and you wind up with other materials having trade-offs. So like to improve a membrane that has improved air exchange will tend to also have more water permeation. So then you wind up losing water from your culture. But if you have the capacity to then add water back as the culture continues to dry out, you could do that as another option. And one other thing we saw at ASGSR was those hollow fiber filters that could also potentially be a solution.
Interesting.
For bionutrients for the ground control, you shook some of them and not others. Is that
Yeah.
right? Okay.
Yeah. So some of those controls were shaken and those grew much better. But they
Yeah,
all
I would
lost
imagine.
that particular membrane on that, so what Rachel's talking about is a bioreactor that looks like this, right? And it had the membrane on one end, right, like on this end, with air holes to exchange. And these guys, I think they lost at least 10%. No, they lost more. They lost like 20% of their volume during the culturing, and that was just 48 hours. Yeah. So as you increase air exchange, the more you lose in water. Yeah. So yeah, I got to think about that trade-off. I mean, that's anybody who works on plants, or agriculture, everything's trade-offs, right? And you got to engineer it on.
No, put that in a box, and it's basic. But then that box you could then have a condensation system that somehow like drains like the air, and then puts that water back into the irrigation system because it's got to be close.
Yeah, or into potable water, right? Because it's transpired or evaporated water. The only downside is that if you have lots of systems that are doing that, lots of plant systems, then all of a sudden your energy requirements start going up for whatever spacecraft you're looking at or whatever habitat, right? Because if you're just processing liters and liters and thousands of liters of water a day, that's a lot of energy and a much larger scale system for condensate for getting the condensates. Yeah, it's a challenge for an integrated eclipse kind of system or bioregenerative eclipse.
And I guess there's no way you could ever produce enough hydrogen as a byproduct to kind of fuel the whole thing of
Yeah, I mean, it's just yeah. So a lot of spaceflight technologies, that's a big chunk of it is like, what's the trade? How much weight and volume do you really need to get your work done? And then does it produce enough to then justify that over doing a camping trip?
Exactly. Yeah, So I was referring to one of the statements from, I think it was Tech Masters, when they were showing what portfolio they had. And one of the things was a biocell that created hydrogen gas. I was like, just trying to think, could that ever really pay for itself in like, but maybe not.
Maybe not. Yeah. Yeah. And is it, are we going from camping trip to cruise ship, or are we actually going to go to something more self-sufficient? Yeah.
Yeah. And that's such a good point, isn't it? So, and that makes me think of like when they were describing just yesterday the Mars plans, different portfolios, like Lava Newman was presenting, and she was saying there's like the 3 30-day missions plan, which is 3 camping trips, and then there was the one which has the target of drilling down to find liquid water, and that one is like the cruise ship approach, where essentially you have to send over a bunch of ships to build some infrastructure to keep people alive for like 300 days, and so it has to be just much more bigger. And so taking us back to the mission profile and plan really dictates what you take with you and what technologies you need, right?
Yep.
And so let's hopefully get cruise ship. Who doesn't want? Oh, if I said who doesn't want a nuclear reactor on the moon, would I be controversial? Who doesn't want lots of power on the moon to get through nighttime? I digress. Anyway, well, this has been awesome, Mark. Thanks for sharing all those like insights and lessons learned, and we've got some insights into your next experiment too. So that was like going beyond, and yeah. So
yeah, let's hope it happens. Let's hope that I'm still hoping for the funding to remain and be able to actually complete, right? Because that's always a hazard with that. With actually any spaceflight work, is that you could run into a hiccup that you just can't get the experiment done.
Yeah,
that's why flight opportunities are really rare and valuable. Yeah.
Yeah, very well put.
Yeah.
In these strange times when it's just like anything can suddenly get cut, particularly when you like Viper, Mars sample return.
Yeah, I mean Viper's an excellent example, but I mean you can also see why, because there was like the cost benefit and risk benefit. It just got out of whack for that CLIPS lander, because it was a group that had trouble with their first lander, and then they were trying to do something much larger scale for their second to deliver Viper, and it was just like, okay, this is too valuable of a payload to risk on an unproven such an unproven delivery technology. Yeah.
So yeah, flight readiness ready. We referred to it briefly earlier to, I think the gentleman's gone, but yeah, it makes sense, right? Showing this is going to work. This is so important. Well, once again, thanks everyone for coming out. Our time has like come to an end, but it's clear that we're all enjoying the science and this lucky goal that we all have, which is to somehow create the foundations of space agriculture, because it's multifaceted endeavor, and as we see, it's not just flowering plants are going to be part of this equation. It's the single cell like cousins and lots of other like technologies that need to come together to make this possible. Nick's thinking, don't forget the microbes.
Well, and don't forget the mushrooms and things like that too, right? Because you need for that full closed loop, anything that you can do to reduce the amount of material that needs to then go, say, into a black water stream, that would be really helpful, right? It can be, yeah.
So like, I have to go to lab today because the student over the weekend, they took a mushroom, they put electrodes in it, they then put it in a glass jar, they put it on the back of one of those 4-legged robots, those like guard dog thingies. And then I've seen the video of it walking in and out of the shadow as it tried to optimize its temperature inside. The mushroom was sending signals to the robot that was moving into the shadow to cool it down and out to warm it up. So I've got to go see what else this Oliver gentleman has done with this mushroom in a jar. It really does remind me of like Futurama, the head in the jar kind of floating around. And I'll bring the videos for you guys to like see and laugh at in the next meeting. But you mentioned mushroom, you fell into the trap, as it were, like funny word today. Yes, quite. So yes, real pleasure. Thanks again, everyone, for like coming to hang out, and make sure there's some real great science again in the new year. So, Merry Christmas, seasons, greetings, Happy Hanukkah, whatever it is you like to celebrate around this time of year. Make sure you eat and drink too much of your loved ones, and yes, reach out if you need anything, and we'll see you in the new years. Okay.
Cheers.
Thanks, everyone. See you guys.
Yeah.
Bye.